In this lecture, we're going to look at molecular techniques that are used in the laboratory to study viruses. These techniques can be put into three basic categories: concentration and purification of viruses, characterizing virus genomes, and studying virus proteins and messenger RNA. So we'll look at some of the more commonly used protocols in this lecture. Ultracentrifugation is a specialized technique used to spin samples at exceptionally high speeds. The basis of ultracentrifugation is the same as normal centrifugation, to separate the components of a solution based on their size, density, and the density of the medium. Preparative centrifugation leads to separation, isolation, and purification of various cellular constituents in viruses. And it's important to note that viruses are so tiny that they will not pellet out using a standard benchtop centrifuge. In fact, we'll use standard centrifugation to clean up the cell culture medium that we used for growing virus, to clear out the cellular debris left from the destruction of the cells by the virus during its replication. And we get a clean supernatant that is free of cellular debris but contains the virus. Differential centrifugation is used to separate the components of a solution based on differences in the sedimentation rate of the different components of the mixture. Density gradient centrifugation goes further in particle separation than differential centrifugation. It is ideal when the goal is to isolate particles of similar sizes but different densities. Rate zonal centrifugation is one type of density gradient centrifugation. In this case, you layer a sample on top of a density gradient, so you preform the gradient. The particles will separate into bands or zones with characteristic sedimentation rate. This depends on size, shape, density, centrifugal force, and density of the gradient. Sucrose is very commonly used in rate zonal centrifugation because it forms sharp, easily isolated bands of pure virus. However, it can be very viscous at high density. Isopycnic centrifugation separates particles solely on the basis of different densities. Cesium chloride is very soluble and high density and has a low ionic strength. It does not absorb UV light or interfere with scintillation fluids, and it can be removed from the virus fraction by dialysis or filtration. So cesium chloride is very commonly used with isopycnic centrifugation. But let's look at these a little bit more closely. Remember, in differential centrifugation, the separation of various components is achieved primarily based on the size of the particles. It is commonly used in simple pelleting and in obtaining partially pure preparation of subcellular organelles and macromolecules. The separation process is based on size and density of the particles. Larger and denser particles get pelleted out at lower centrifugal forces. So, for example, unbroken whole cells will pellet at low speeds and short intervals, such as 1000 G for 5 minutes. Smaller cell fragments and organelles remain in the supernatant and require more force and time to get pelleted out. Depending on their separating order, cells can be derived for further application. Homogenation of the tissue sample is the first step before doing centrifugation. The tissue is homogenized in a blender to break the cell membrane and mix all the cell constituents. The mixture obtained after homogenation is called the homogenate. Samples are placed in tubes within or attached to the rotor in the centrifuge. The speed of rotation is increased at each stage that leads to the separation of cellular components. Different fragments of cells have different sizes and density. Thus, each fragment settles into a pellet with different minimum centrifugal forces. So you can see here in this illustration, first you homogenize, you take that homogenate, and you start doing differential centrifugation. So at each step, you increase the force that's being used, you increase the time, and different components will pellet out. So in this case, the final pellet that's obtained would be the ribosomes. Here we see an example of rate zonal centrifugation using a sucrose gradient. So remember, the sucrose gradient is preformed and it increases from top to bottom. You layer your solution on top of this gradient and centrifuge at high G-force for several days. The particles sediment downward until they reach a layer with density equivalent to their own. The gradient is separated into fractions, which are analyzed for the presence of virus. And keep in mind, I said that they can be centrifuged for days. That's a key part of working with ultracentrifuges. You're often running these you're doing a run with an ultracentrifuge that can last for hours or even for days. So again, this is very different than normal centrifugation. But you run your rate zonal centrifugation, your particles separate out in the tube according to their density. And then you can retrieve the fractions simply by putting a small hole in the bottom of the tube. So you go through the collection of all these fractions, and then you can do various kinds of tests to see where the particle that you're looking for has been retrieved. Due to the importance and increasing use of PCR in detection and diagnosis, I want to focus on that procedure for a minute or two. PCR is a simple, sensitive, and versatile technique that can provide results rapidly. This method is used to create or amplify huge numbers of copies of a specific DNA fragment. Because both strands are copied, there is an exponential increase in the number of copies of the gene. As the story goes, the concept of PCR was developed by Kary Mullis during a moonlit drive through the California mountains while experiencing the effects of LSD. He later won the Nobel Prize for his discovery. Ingredients needed to copy DNA include the template or target DNA or a complementary DNA copy of RNA made with reverse transcriptase, sequence-specific primers, deoxynucleotide triphosphate building blocks, and a thermostable polymerase with an appropriate buffer. Design of primers is crucial in order to ensure specificity. Several thermostable polymerases are now on the market. Taq DNA polymerase was isolated from the bacterium Thermus aquaticus from hot springs in Yellowstone National Park. It has a drawback in that it lacks proofreading activity and thus produces errors every 10 to the 4th or 10 to the 5th bases. Other polymerases, such as PFU, that was isolated from Pyrococcus furiosus, contain the proofreading ability. The ingredients are placed in a tube and placed in the thermocycler. There are 3 major steps in a PCR, which are repeated for 30 to 40 cycles, and they include denaturation at 90 to 95 degrees centigrade to separate or melt the double-stranded DNA into single strands, annealing at temperature close to the melting temperature of the primers, and extension or polymeration, usually at 72 degrees centigrade, which is the ideal working temperature for the polymerase. There is only one copy of the wanted gene before the cycling starts. After one cycle, there will be 2 copies. After 2 cycles, there will be 4 copies, and so on. PCR can be used to detect some viruses as is. These viruses have a genome that is made of DNA, but many viruses don't store their genetic code as DNA. They use RNA as the plan from which they make more of themselves and their viral proteins. In these cases, DNA only plays an intermediate role, if any. DNA viruses include the adenoviruses, herpes viruses, polyomaviruses, parvoviruses, and so on. PCR also works well for plasmids and human genes and other DNA fragments we want to work with in the lab for reasons other than detecting a virus in a human. RNA viruses include influenza viruses, parainfluenza viruses, rhinoviruses, coronaviruses, Zika viruses, and so on. PCR is based around the use of a heat-stable DNA-dependent DNA polymerase enzyme. For the PCR to work, we need to first make virus RNA genes or genomes into DNA so that the main enzyme can use it and duplicate it and make enough of it to detect or use in molecular biology, or whatever the downstream application may be. To make a DNA copy of the RNA, we need to add in another enzyme and another preceding step to the PCR process. That enzyme, the reverse transcriptase, or RT, is added in a step called reverse transcription. The addition of the RT step changes the naming technique from PCR to RT-PCR. In order to verify that a gene was copied and is the right size, an agarose gel can be run. A DNA ladder, which is a mixture of fragments with known size, can be loaded to compare with the PCR fragments. DNA which is negatively charged due to the phosphate groups will move from the negative position, where the sample is loaded, to the positive pole due to the current being passed through the gel. The agarose is a matrix with pores which allows DNA of various sizes to run differently through the gel. Shorter molecules move faster than longer molecules, and thus the DNA can be separated. Ideally, one would look for the formation of a single band at the estimated size of the DNA fragment. PCR products can be used as a target for hybridization, for direct sequencing of the DNA, and as a specific probe. PCR has several important advantages. Because the pathogen does not need to be cultured before detection, it is possible to detect a single pathogen or many related pathogens in less time and more cheaply. This procedure is useful for detection of animal and plant viruses and is being used more and more for fungal and bacterial plant pathogens in our disease and diagnostic clinics. In conventional PCR, the amplified product is detected by an endpoint analysis, such as running the DNA on an agarose gel after the reaction is finished. In contrast, real-time PCR, as the name suggests, measures the amount of PCR product at the end of each amplification cycle, thus in real time. To do this, you label the PCR products during or after the primer extension step of each amplification cycle. Real-time detection is made possible by including fluorescent molecules in the reaction that report an increase in the amount of DNA with a proportional increase in fluorescent signal. These molecules include DNA binding dyes like cybergreen and fluorescently labeled sequence-specific primers like TACMAN. Figure 1 shows what happens to a fluorogenic probe during the extension phase of PCR. If the target sequence is present, the probe anneals downstream from one of the primer sites and is cleaved by the 5' nuclease activity of Taq DNA polymerase as this primer is extended. This cleavage of the probe separates the reporter dye from quencher dye, increasing report dye signal. Cleavage removes the probe from the target strand, allowing primer extension to continue to the end of the template strand. Figure 2 shows a representative amplification plot and defines the terms used in the quantification analysis. An amplification plot is the plot of fluorescent signal versus cycle number. In the initial cycles of PCR, there is little change in fluorescent signal. This defines the baseline. An increase in fluorescence above the baseline indicates the detection of accumulated PCR product. A fixed fluorescent threshold can be set above baseline. The threshold cycle of CT is defined as the fractional cycle number at which the fluorescence passes the fixed threshold. A plot of the log of initial target copy number for a set of standards versus CT is a straight line. The main advantage over conventional PCR is that it allows you to determine the starting template copy number. It can either be qualitative, that is, presence or absence of sequence, or quantitative, as in the number of copies of DNA. Real-time PCR data can be evaluated without gel electrophoresis, resulting in reduced experiment time and increased throughput. Since everything can be evaluated in a closed system, opportunities for contamination are reduced and the need for running a gel is eliminated. The downside is that the equipment needed is expensive and experienced technicians are required to perform the analysis. It is also important to standardize the procedures across all labs to ensure accuracy of diagnoses. DNA sequencing is the process of determining the nucleic acid sequence, the order of nucleotides, in DNA. It includes any method or technology that is used to determine the order of the 4 bases, adenine, guanine, cytosine, and thymine. The advent of rapid DNA sequencing methods has greatly accelerated biological and medical research and discovery. Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain-terminating dideoxynucleotides by DNA polymerase. A chain terminator lacks a 3' OH for the attachment of the next nucleotide. The classical chain termination method requires a single-stranded DNA template, a DNA primer, a DNA polymerase, normal deoxynucleotide triphosphates, or DNTPs, and modified dideoxynucleotide triphosphates, or DDNTPs, the latter of which terminate the DNA strand elongation. These chain-terminating nucleotides lack the OH group required for the formation of the phosphodiester bond between 2 nucleotides, causing DNA polymerase to cease extension of DNA when a modified DDNTP is incorporated. The DDNTPs may be radioactively or fluorescently labeled for detection in automated sequencing machines. Most sequencing is now automated. Instead of labeling fragments with radioactive primers, modern sequencing machines use fluorescent dyes to tag the DDNTs, one color for each of the 4 types of nucleotides. This allows for all 4 reactions to be done in the same tube. Sensors measure the wavelength of the overlapping fragments of DNA in the reaction mixture, and it's separated by electrophoresis and moved past the detecting sites. The results are recorded and stored in a database for later sequence interpretation. So, how does viral genome sequencing help? Why is it so widely used in laboratories now? I thought that it would be worthwhile just to consider how valuable virus genome sequencing has been in our understanding of COVID-19. So remember, virus genome sequencing is a vital and rapidly developing tool, and it's being used in the diagnosis of COVID-19 and in understanding the spread and control of the new virus. So how do we use viral genome sequencing? Well, first of all, genomes identify viruses. If the new coronavirus's sequence is found in a sample, usually taken from the nose or the mouth, it will confirm the likelihood that a patient's symptoms are those of COVID-19. Virus genomes constantly alter or mutate, changing a few letters at a time as they divide and spread by infecting more people. These changes can be exploited to track the spread of the virus by sequencing, recording, and analyzing genomes. If the virus genome sequencing is undertaken rapidly on a large scale, then it can assist epidemiologists and public health authorities in understanding how the virus is spreading and evaluating how effective their interventions have been. It can also help to establish whether new variants are associated with a particular pattern of symptoms or severity of disease. In the longer term, tracking new variants is likely to be extremely important to ensure that vaccines, when these are developed, can be kept up to date with the strains of virus that are currently circulating. We can also use it in terms of studying local transmission of virus as opposed to imported cases. In the initial stages of the epidemic, sequencing can be used to find out how many new cases of disease are imported or come from local transmission. Global databases of virus genomes enable researchers to compare genomes so that an accurate assessment of local transmission in each country can be made. Virus genome sequencing is used to look at epidemic growth. Mathematical models of how viruses evolve during an epidemic, developed from extensive analysis of past outbreaks, allow epidemic growth rates and other measurements of transmission and infection to be estimated from virus genome sequences. Compared to the estimates from other sources of data, insights from virus genetics are most useful for the prediction of longer-term, longer-scale trends. Importantly, they provide independent validation of estimates of the size and growth rate of an epidemic. This is useful especially when cases are underreported, for instance because many people who are infected do not have symptoms. You can use it to look at spread in different places or groups. Widespread sampling and genome sequencing of the new coronavirus allows the construction of virus spread in different places or groups of people. This provides information about what is driving the spread of virus, both locally and nationally. This work can be made more precise if virus genomes are combined with information about where, how, and when people travel locally and internationally. It can be used to look at transmission chains. Virus genome sequences can also identify unique genetic changes shared by all those infected in a single virus transmission chain. This can be used to distinguish whether two clusters of cases in the same area have arisen because one started infection in the other, or because there were two distinct and independent chains of transmission with separate earlier origins. Virus genomes can therefore add to the information provided by patient contact tracing, which is important for tracking outbreaks in communities, hospitals, and other care settings. And then genetic variation. Many genetic changes that occur in the genome of the virus will have no significant effect on the course of infection or disease, or the impact of control measures. However, a few of the changes might be important. These need to be identified and tracked through time. In viruses such as influenza, we know that genetic changes can alter how the immune system recognizes virus, resistance to antiviral drugs, and the severity of disease. These discoveries have yet to be made for the new coronavirus. Rapid, large-scale virus genome sequencing is a new stream of information that can contribute to the tracking of epidemics and the development of new methods of control. Its application to the new coronavirus is only just beginning. A microarray is a laboratory tool used to detect the expression of thousands of genes at the same time. DNA microarrays are microscope slides that are printed with thousands of tiny spots in defined positions, with each spot containing a known DNA sequence or gene. Often these slides are referred to as gene chips or DNA chips. The DNA molecules attached to each slide act as probes to detect gene expression, which is also known as the transcriptome, or the set of messenger RNA, mRNA transcripts, expressed by a group of genes. To perform a microarray analysis, mRNA molecules are typically collected from both an experimental sample and a reference sample. For example, the reference sample could be collected from a healthy individual, and the experimental sample could be collected from an individual with a disease. The two mRNA samples are then converted into complementary DNA, or cDNA, and each sample is labeled with a fluorescent probe of a different color. For instance, the experimental cDNA sample may be labeled with a red fluorescent dye, whereas the reference cDNA may be labeled with a green fluorescent dye. The two samples are then mixed together and allowed to bind to the microarray slide. The process in which the cDNA molecules bind to the DNA probes on the slide is called hybridization. Following hybridization, the microarray is scanned to measure the expression of each gene printed on the slide. If the expression of a particular gene is higher in the experimental sample than in the reference sample, then the corresponding spot on the microarray would appear red. In contrast, if the expression in the experimental sample is lower than in the reference sample, then the spot would appear green. Finally, if there is equal expression in the two samples, then the spot would appear yellow. The data gathered through microarrays can be used to create gene expression profiles, which show simultaneous changes in the expression of many genes in response to a particular condition or treatment. Microchips have been used extensively to study viral biology, many times addressing ecologically relevant questions. Most of these studies are circumscribed to the analysis of isolated viruses or to issues related to the role of viruses in human health. Microarrays are created by the immobilization of molecules in discrete spatial locations on a solid surface. Depending on the nature of the molecule immobilized on the physical substrate, there are different types of microarrays such as DNA, protein, glycan, small molecule, cell, and even tissues. The interaction between immobilized probes and targets is normally detected by fluorescence, although other detection systems are also available. The power of microarrays lies in their unprecedented capacity to simultaneously interrogate tens to hundreds of thousands of immobilized probes. Microarrays have been used for studying emergent pathogens. This is shown on this slide looking at severe acute respiratory syndrome, or SARS, and avian influenza virus, H5N1. Various approaches have been developed for detection and diagnostics with DNA and protein microarrays. In A, you see specific DNA probes, for instance, COVID, orange, for SARS, and H5, green, for H5N1, that can be used to detect the presence of pathogen DNA or RNA using PCR or reverse transcriptase, thus enabling multiple pathogens to be detected simultaneously. Tiling arrays can be used to sequence pathogens so that any mutations in evolving pathogens can be rapidly detected and tracked. As illustrated in B, 4 probes, one for each nucleotide base, are used to determine the genotype at a given loci. A set of probes designed for an arbitrary position XYZ is shown to reveal the unknown base to be adenine. Multiple probe sets are tiled across the whole pathogen genome and, upon hybridization and analysis, can confer the complete sequence information of the pathogen with great accuracy. And then in C, you can see antibody arrays make use of the sandwich immunoassay to screen for the presence of a pathogen. As depicted, the pathogen sample is first applied to the microarray, followed by the reporter antibody. As a result, the pathogen is sandwiched in between 2 antibodies, an immobilized antibody blue and a tagged antibody purple, capable of reporting the presence of a pathogen on a microarray. And then down in D, you can see proteome microarrays, which contain immobilized proteins from the target pathogen, which can be screened against sera obtained from infected individuals. If the individual has been exposed to the pathogen, the sera will contain the antibodies, blue, against specific antigens of the pathogens that will react with the immobilized protein that can be detected using tagged secondary antibodies, purple. This procedure also facilitates the identification of specific immunodominant antigens present in the pathogen proteome. These proteins are considered to be largely responsible for triggering the host immune response and thus have high potential for the development of vaccines against particular pathogens. Here we see a microarray that was designed for screening dengue virus samples. This DNA microarray assay is specific and sensitive and can detect dual infections with 2 dengue virus serotypes and single serotype infections. The thing to keep in mind with dengue is that there are 4 serotypes, and individuals are capable of becoming infected with all 4 serotypes. So this particular microarray can detect all 4 of those. Other methodologies may underestimate samples containing more than 1 serotype. This technology can be used to discriminate between the 4 serotypes. Single-stranded DNA targets were covalently attached to glass slides and hybridized with specific labeled probes. The dengue virus isolates and dengue samples were used to evaluate microarray performance. The results demonstrate that the probes hybridized specifically to dengue virus serotypes, with no detection of unspecific signals. This finding provides evidence that specific probes can effectively identify single and double infections in dengue virus samples. The ViroChip is a pan-viral microarray designed to simultaneously detect all known viruses as well as novel viruses on the basis of conserved sequence homology. In the slide, it demonstrates how to run a ViroChip assay to analyze clinical samples for the presence of both known and unknown viruses. The steps of the ViroChip assay include nucleic acid extraction from clinical samples, reverse transcription of extracted RNA and second-strand cDNA synthesis, PCR amplification of randomly primed cDNA, fluorescent dye incorporation, hybridization of labeled material to the virochip microarray, and finally scanning and analysis. This shows individual virus patterns on a diagnostic DNA microarray. Each probe is printed on a subgrid in quadruplicate and forms a distinctive pattern as shown by shaded squares. In turn, subgrids are arranged in larger units, metagrids, which also create a unique virus-specific pattern on a slide. The crosses in the middle of each subgrid indicate blank spaces. Now here's something that's pretty cool. New technology developed by Howard Hughes Medical Institute researchers makes it possible to test for current and past infections with any known human virus by analyzing a single drop of the person's blood. The method, called VirScan, is an efficient alternative to existing diagnostics that test for specific viruses one at a time. With VirScan, scientists can run a single test to determine which viruses have infected an individual rather than limiting their analysis to particular viruses. That unbiased approach could uncover unexpected factors affecting individual patients' health and also expands opportunities to analyze and compare viral infections in large populations. The comprehensive analysis can be performed for about $25 per blood sample. The test is currently being used only as a research tool and is not commercially available. VirScan combines DNA microarray synthesis and bacteriophage display to create a uniform, synthetic representation of peptide epitopes comprising the human virome. Immunoprecipitation and high-throughput DNA sequencing reveal the peptides recognized by antibodies in the sample. The color of each cell in the heat map depicts the relative number of antigenic epitopes detected for a virus, rows, in each sample, which would be the columns. So, we have looked at how to concentrate and purify viruses using ultracentrifugation, and we've also looked at several methods for studying virus genomes. Now we're going to discuss ways to study viral proteins and viral messenger RNA. One of these methods is pulse labeling. Pulse labeling is a technique in which radioisotopes are used to measure the rates of synthesis of molecules within living cells. A suspension of cells or organelles is exposed to a small quantity of an isotope for a brief period, just seconds or possibly minutes, hence the term pulse. The medium containing the isotope is then removed. There can be a problem with detecting viral proteins against the background of cellular proteins using standard methods. However, many viral infections lead to partial or total shutoff of host cell messenger RNA or protein synthesis, while viral proteins and messenger RNA are synthesized at high rates. And the illustration you see here, on the left gel, we have HeLa cells that were infected with poliovirus and labeled with S35 for 2-hour pulses. Remember, S35 is going to be taken up by proteins. Protein is isolated and fractionated, then localized by autoradiography. Many cellular proteins are shut off, indicated by the O, while those that continue to be synthesized are marked with the O and an asterisk. On the right, you see viro cells that were infected with herpes simplex 1. Viral proteins synthesized early in infection are indicated by E. L indicates late proteins. Cellular proteins are rapidly shut off and are indicated by C. C asterisk marks proteins that do not appear to be shut off or whose synthesis increases after infection. A pulse chase experiment is a two-phase technique used to examine cellular processes that take place over a period of time. During the pulse phase of the experiment, cells are exposed to a labeled compound. The labeled compound is incorporated into the molecule or pathway being studied. In the chase phase, an unlabeled form replaces the labeled compound. The reaction is monitored to see how long it takes the labeled form of the compound to be replaced by the unlabeled form. There are many ways to label a compound for use in a pulse chase experiment. Radioisotopes or fluorescent labeling using compounds such as green fluorescent protein are both popular. But in this gel that you see in the slide, an early pulse was used, and then they followed it over time. You can see at the top the hours. And you start off with one large labeled band. Remember, bands up at the top are going to be your largest proteins. Smaller proteins will migrate further down the gel. Now, over time, notice what happens. That large band disappears, and you get the appearance of many small bands. We'll talk more about this later when we go through poliovirus in the later lectures. But what's happening is that a large polyprotein is first produced. That's what you see in that big band. And then over time, that polyprotein is cleaved into smaller proteins. And that's why you see the large band disappearing and the smaller bands starting to show up. Antibodies are specialized Y-shaped proteins that bind like a lock and key to the body's foreign invaders, whether they are viruses, bacteria, fungi, or parasites. Antibodies are part of the adaptive immune system, the arm of the immune system that learns to recognize and eliminate specific pathogens. Antibodies are made by plasma cells, a type of white blood cell, in response to an antigen, and each antibody can bind to only one specific antigen. We'll talk more about antibodies as a part of the immune system in another lecture. However, due to their high specificity and selectivity, antibodies have always had the potential to be of great use as biochemical tools for a range of applications, including selection, identification, purification, and as therapeutics and in diagnostics. Routine procedures such as Western blot, flow cytometry, immunohistochemistry, enzyme-linked immunoassays, and many others all rely on antibodies. Broadly speaking, antibodies can be categorized into two groups, polyclonal or monoclonal, and they're utilized in three main areas: research, diagnostics, and therapeutics. Antibodies, also known as immunoglobulins, are secreted by B cells or plasma cells to neutralize antigens such as bacteria and viruses. The classical representation of an antibody is a Y-shaped molecule composed of 4 polypeptides, 2 heavy chains and 2 light chains. Each tip of the Y contains a paratope that is specific for one particular epitope on an antigen, allowing these two structures to bind together with precision. The ability of binding to an antigen has led to their ubiquitous use in a variety of life science and medical science applications. These antibodies can be classified into 2 primary types, monoclonal and polyclonal, by the means in which they are created from lymphocytes. Each of them has an important role in the immune system, diagnostic exams, and treatments. Polyclonal antibodies are a heterogeneous mixture produced by different B cell clones in the body. They can recognize and bind to many different epitopes of a single antigen. Polyclonal antibodies are produced by injecting an immunogen into an animal. After being injected with a specific antigen to elicit a primary immune response, the animal is given a secondary, even tertiary immunization to produce higher levels of antibodies against a particular antigen. After immunization, polyclonal antibodies can be obtained straight from the serum or purified to obtain a solution which is free from other serum proteins. Monoclonal antibodies are generated by identical B cells, which are clones from a single parent cell. This means that the monoclonal antibodies have monovalent affinity and only recognize the same single epitope of an antigen. Unlike polyclonal antibodies, which are produced in live animals, monoclonal antibodies are produced ex vivo using tissue culture techniques. The process begins with an injection of the desired antigen into an animal, often a mouse, multiple times. Once the animal develops an immune response, the B lymphocytes are isolated from the animal's spleen and fused with myeloma cell lines, creating immortalized B cell myeloma hybridomas. The hybridomas, which are able to grow continuously in culture while producing antibodies, are then screened for the desired antibody. In this slide, you see monoclonal and polyclonal antibodies binding with the same antigen. I'm often asked which is better, a monoclonal or polyclonal antibody. Well, it depends on what you plan to use it for. Polyclonal antibodies are ideal reagents in diagnostic assays and hemagglutination reactions due to their ability to recognize different epitopes of a target molecule. The best use of polyclonal antibodies is to detect unknown antigens. Polyclonal antibodies are used as a secondary antibody in immunoassays such as ELISAs. Their role is to bind to different epitopes and amplify the signal, leading to better detection. Monoclonal antibodies, in contrast, provide an unlimited source of antibody that is homogeneous and, once characterized, predictable in its behavior. Monoclonal antibodies are often used as primary antibodies in immunoassays due to their ability to bind specifically to a single epitope of an antigen. Monoclonal antibodies have also been very useful in the development of treatments because of their ability to find and target specific molecules. The monoclonal antibodies' Fc regions can be tagged with markers that can then be used to discover cellular surface components. More than 40 monoclonal antibodies have been approved by the FDA to fight cancers, rheumatoid arthritis, asthma, and other illnesses. Monoclonal antibodies are also being used as vectors to bring drugs to target cells. When the antibody-drug conjugates meet the target cell, the drug is released and exerts its effect. Okay, now that we've gone through all these different virus methods, to finish up this module, what I want to do is go through an actual research paper and talk about how all these different methods can be used to tell a story, and that's essentially what a published paper is. And then lastly, talk about the different kinds of testing that's currently being done for COVID. Okay, so first, the research paper. This paper was published in Emerging Infectious Diseases, and it was a group of spunky medical entomologists from here at Virginia Tech. Now, just take a moment, stop typing, stop writing, just listen to this, to what I'm going to tell you. I'm not going to test you on this. I'm not that vain that I would make you study one of my papers. I'm just using it as an example of how you have to utilize so many different techniques in order to get a full project completed. So for this paper, we were looking at Cache Valley virus, which is an arthropod-borne virus in Aedes japonicus mosquitoes. Aedes japonicus mosquitoes are a newly introduced species. They're exotics, and they've only been in the U.S. for 2 decades, I guess. So they've been spreading, and as they've moved into new areas in the country, they've gotten involved in some of the epidemiological cycles of the already existing arthropod-borne diseases, and that's what we saw with Cache Valley. So we published this paper, and I'm going to take you through the steps. Okay, so these are the steps that we had to go through in order to do this particular project. So the first thing, of course, is we have to collect the mosquitoes from the field. And to do this, we used gravit traps. The way these traps work is that black container at the bottom, we would put hay infusion in there. That's just water with some hay that's been allowed to ferment a little bit so that it gives off volatiles that are attractive to mosquitoes. So the mosquitoes will come in wanting to lay their eggs. But above the water surface, you can see that little black cylinder. That's a fan. So when the mosquitoes come in, they get sucked up, and then they're held in that net. And we can take them back to the lab. And once we have them in the lab, we're going to sort them. So here you can see we freeze the mosquitoes down, then keeping them on ice, we will sort them and look at them based on morphology to identify them to species. We separate them by sex, by trap number, by date. So ultimately what we're going to want to do is to form pools of mosquitoes. And so here you can see we're putting a mosquito into a tube. Well, we collected hundreds of mosquitoes during this project, and you can't afford to analyze each one separately. So we put them into groups in the tubes, but we'll put only the same species in the tubes. We'll again separate them by trap number and date and so on. And we ended up with, even with pooling, we had over 400 pools of Aedes japonicus. So that was still a lot of samples for us to have to analyze. But this pooling of your specimens is a standard part of doing any kind of research like this involving collecting mosquitoes in the field. Okay, so next thing we do, once we've got the mosquitoes separated into pools and all that, we want to move on to do testing for virus. So the next step is grinding the mosquitoes for virus testing. And here you can see in these tubes down at the bottom are those little shiny things at the bottom of the tubes are BBs. So we put cell culture medium and BBs in the tubes with the mosquitoes and just hold them on a vortex mixer so that the mosquitoes become completely, essentially disintegrated. And then what we can do is take these tubes and put them in a standard benchtop centrifuge and spin them down. And what that will do is pellet out all of the mosquito parts. But the virus will remain in the supernatant, because remember, you have to use an ultracentrifuge to pellet out virus. So we can spin the samples down and clean them up, get rid of all the mosquito debris and so on, and just work with the supernatant, which will have the virus. So then we want to screen for the virus itself, and we were going to use a infectivity assay, one that's based on CPE. In this case, we were just going to do an all or none test in 96 well plates. So each one of our samples was inoculated into a 96 well plate, and we just looked for positives or negatives. Remember, where you see the solid dark blue, those would be negative wells. The blue is the viable cells that have taken up the crystal violet. But you can see several of the wells in this illustration appear to be positive. But what we would want to do next is confirm that the CPE was actually caused by virus, because there are many things that can cause cells to die off, various kinds of toxins and things. So we wanted to confirm that it was actually virus, so we did plaque assays. But we were able to do plaque assays just on the putative positive samples, not on all 400 plus samples. And so that's what we did. And when you see these plaques, that's a confirmation that the pathogen that you're working with is actually a virus. Okay, so now we've collected mosquitoes, we've isolated virus from these mosquitoes, but now we have to confirm what virus it is. We have to identify the virus and then do molecular analysis of the virus. And we did this using several methods. We were able to amplify the virus isolates on viro cells. Then we extracted virus RNA from infected cell supernatants using the QAMP viral mini prep kit. We used reverse transcription PCR and a bunyavirity specific universal primer to produce amplicons of the small RNA segment, which was sequenced. Then the sequencing, we sent this out for sequencing, and then a blast query indicated that the isolates were Cache Valley virus. We then amplified the large RNA segments by using reverse transcription PCR and Cache Valley virus specific primers, and amplified the medium RNA segments as well. The resulting amplicons were sequenced and identified as Cache Valley virus, again by using a blast query. The medium RNA segments, and it's the medium RNA segment of a bunyavirus which encodes the viral glycoprotein. These segments were amplified and then sequenced. We used the medium RNA segment then to infer phylogeny with other Cache Valley virus isolates that were reported by another researcher at a different institution. We translated the RNA sequences to amino acid sequences and aligned them visually. And this is the phylogeny that we ended up with. So down here, down at the bottom, you can see that I indicated in a gray shading are two Cache Valley virus isolates, and you can see that they fall out into lineage 2. And lineage 2 is a lineage of Cache Valley virus that is thought to have originated down in Mexico and then has been moving northward over the past couple of decades. Okay, so again, this was just to show you that just to do one research paper, you have to employ a lot of different methods, a lot of different techniques. Okay, so now I want to shift gears and talk about how we use some of the tests that we talked about for diagnostics. If you've learned one thing about laboratory testing for COVID-19 in the past couple of months, it's probably that it's complicated. If you've been paying close attention, you might also know that it's not a one-size-fits-all approach. As new tools like antibody and rapid antigen testing became readily available, new questions continued to arise. It's important that we understand the value and limitations of COVID-19 testing so that we can best use the current and developing items in our toolbox to effectively control the spread of COVID-19. There are two basic types of tests: diagnostic tests and antibody tests. A diagnostic test can show if you have an active coronavirus infection and should take steps to quarantine or isolate yourself from others. Currently, there are two types of diagnostic tests which detect the virus: molecular tests, such as RT-PCR tests, that detect the virus genetic material, and antigen tests that detect specific proteins on the surface of the virus. An antibody test looks for antibodies that are made by your immune system in response to a threat, such as a specific virus. Antibodies can help fight infections, but antibodies can take several days or weeks to develop after you have an infection. The advantage, though, is that they may stay in your blood for several weeks or more after recovery. Because of this, antibody tests can't be used to diagnose an active coronavirus infection. At this time, researchers do not know if the presence of antibodies means whether or not you're immune to the virus in the future, but at least it tells us whether or not you've been infected. Nucleic acid amplification testing requires respiratory samples from the patient because, again, COVID-19 is a respiratory virus. Nasopharyngeal swabs are most commonly used. Lower respiratory secretions, such as sputum, are also used if a patient has pneumonia or lung involvement with infection. Samples are then processed and tested for COVID-19. The test includes extraction of RNA from the patient specimen, conversion to DNA and PCR amplification with COVID-19 specific primers. Nucleic acid amplification tests tell us if a patient is actively infected with COVID-19. They detect the presence of characteristic sequences of viral genetic material, RNA, in respiratory samples of patients. If the viral RNA is detected, it suggests that the virus might be present. If you test positive for COVID-19, you should self-isolate. Call your doctor if your symptoms worsen, and seek medical help immediately if your condition becomes critical. Molecular tests can yield a false negative result if the level of viral RNA in a particular sample is too low for detection, and results can be skewed if steps are not taken to ensure that the tests are performing properly in the first place. So there's rapid antigen testing. These are the point of care tests. Rapid antigen tests are immunoassays that require respiratory specimens, typically nasopharyngeal or nasal swab, from the patient because COVID-19 is a respiratory virus. Once collected, samples are placed in an extraction buffer or reagent and tested for the presence of COVID-19 specific antigens. The rapid antigen test tells us if a patient is actively infected with COVID-19. They detect the presence of virus-specific proteins called antigens from patient specimens. As of August 18, 2020, the FDA has issued emergency use authorization for 3 rapid antigen tests. All of these detect COVID-19 nucleocapsid protein antigens. Results are produced quickly, generally within 15 minutes, and can be performed near the place and time of patient care, as opposed to having to send the sample to a separate laboratory setting. If the viral antigen is detected, it indicates that the virus is present. These tests are generally less sensitive than molecular tests, and it's important to be aware that there is an increased possibility of false negatives with rapid antigen testing. They are most accurate when viral loads are high, which would usually be within the first couple of symptomatic days. And your healthcare provider may order a molecular test to confirm the results of your rapid antigen test if it indicates that you are negative for COVID-19. If your rapid antigen test is positive, there is no need to confirm with the molecular test, as these tests are highly specific. The immune system naturally produces proteins called antibodies in response to infection. Antibodies recognize and bind to foreign molecules called antigens on the surface of pathogens. When an antibody binds to antigen, it seeks to disable the associated pathogen through direct or indirect mechanisms. Blood samples are used as the specimen type for antibody testing. COVID-19 specific antigens are mixed with the patient's blood, and if antibodies to the virus are present in the blood, the two will bind to one another, resulting in a color change that marks the test positive. It's important to recognize that this test measures the amount of antibodies produced in response to COVID-19 infection and does not test for the presence of the virus itself. Therefore, it does not indicate if a person is currently infected. Antibody tells us if a person has been exposed to virus or other infecting microorganism. They detect the presence of antibodies that are produced in response to infection. Antibody tests will also identify if a person has been exposed to COVID-19 and developed immunity against future infections. According to the CDC, it typically takes 1 to 2 weeks for a patient to develop antibodies to COVID-19. Because of this delay, antibody tests are not currently recommended for the diagnosis of acute cases of COVID. Antibody testing instead reveals if a patient's been exposed to the virus, but it doesn't really tell whether or not an active infection occurred, or whether the antibodies produced are the kind that can prevent another infection. Furthermore, it does not reveal how long protective immunity to COVID-19 will last if developed. If we can determine those things, they will help to inform decisions about the safety of returning to normal activities. Antibody tests can yield false negative results if the test is conducted too early and antibodies have not yet developed. They can yield a false positive if antibodies to coronavirus other than COVID-19 are present, such as MERS or SARS. It is therefore important that serology tests are carefully designed to be specific for COVID-19 and are appropriately validated and approved for COVID-19 testing. Broad use of validated serological tests can give a big picture of the number of people who have been infected or exposed to COVID-19 and help to generate surveillance data as we try to control the spread of the virus. The authorization of complete home tests for antibody testing is under discussion by federal authorities and test developers. So here's a slide that will just reinforce what we've already talked about. Remember that nucleic acid tests usually make use of a targeted nucleic acid amplification technique such as PCR. This amplification can take a small amount of viral genetic material in a patient's sample and convert it to large amounts of DNA that can be tested further. Researchers can use a variety of techniques to detect these large amounts of DNA. Because of the targeted amplification step, nucleic acid tests are usually highly specific and very sensitive. They can be used to detect infection early and remain capable of detecting the COVID-19 genome weeks into infection. Yet they can also be time-consuming, and sometimes they also require bulky lab equipment. Now, the antigen tests use antibodies to detect protein components of the COVID-19. These tests can be very rapid, but they're often less sensitive and less specific than nucleic acid tests. Thus, a negative result from an antigen test is not a good indication that a patient is free of infection. For this reason, the WHO does not currently recommend using antigen tests for COVID-19 diagnostic purposes. Nonetheless, many are working on improving antigen tests. More research is needed, but antigen tests should be able to detect the COVID-19 virus around the time of symptom onset. And then lastly, we have the serological or antibody tests. Serological tests are rapid but have relatively low specificity and sometimes give false positives. Indeed, this issue is compounded by the fact that COVID-19 infected individuals produce varying amounts and types of antibodies. Thus, it's not clear what the limit of detection for these tests should be. Serological tests generally detect infection a couple of weeks after symptom onset and beyond. This is because it takes some time for the patients to develop an immune response. Nonetheless, serological tests with known levels of specificity and sensitivity can be incredibly useful to epidemiologists. They can rapidly use these tests on large populations. With the resultant number of positive tests and the known specificity/sensitivity of the test, they can then estimate how many people in a given population have been infected. Such information may be useful when officials decide whether or not to lift social restrictions. Okay, lastly, I want to talk about how samples are taken. In this picture, you can see how the swab is inserted very deep into the nasal cavity to get the sample. That's the way they're going to take a sample from you if you go to one of the testing centers. And these deep nasal swabs are very effective for collecting virus. If you take a anterior nasal swab, in other words, you don't go in as deeply, then that's not going to be as effective. And of course, if people start getting tests that you can take at home and take the sample yourself, more and more of those self-collected samples are going to be anterior, because most of us are not going to want to stick a swab that far up our nose. But an interesting bit of data has come out showing that saliva is very effective for testing for SARS-CoV-2. So research showed that self-collected nasal swabs, when used alone, can miss nearly 15% of infections, probably because people aren't going in deep enough to get a good sample. But when you use saliva samples, it's essentially just as effective as a deep nasal swab, and I think spitting in a tube would be probably preferable to having somebody shove one of those test swabs all the way up your nose. So anyway, I wanted to go through the different kinds of COVID tests that are out there and what they mean and so on, because I think this is information you should have, and it's impacting your present life, and it's impacting the people around you and people you care about.